University of Minnesota. Gretchen studies freshwater systems and how they're changing, particularly due to environmental changes, and so I'm sure we're going to hear about some of that today. Gretchen has spent some time here in her past, both for her undergraduate degree and her PhD, and then snuck over to Michigan State for the master's degree in between. I think everybody has to do one in between, right? So I don't think there's and has also spent time working at both the Wisconsin and the Minnesota Departments of Natural Resources. So very connected to management and those types of agencies. So thank you very much for being here, and I'll turn it over to you.
Yeah, thanks. Thanks to everybody for being here. I see a lot of familiar faces and people from past life in Wisconsin. So I'm going to talk today about a project that is really like probably 4 projects combined into one, and work that people in my lab and other collaborators have been doing over the past about, well, I guess decade when you bring in all this stuff I'm going to talk about. So yeah, thanks for being here. See if this thing works.
Otherwise, I have one.
It works like this, so that's good. Okay, so I'm just starting with one of my favorite things to go down a rabbit hole in is with using ChatGPT to make images for me. And so I gave it the abstract of my talk and told it to make a title slide, so this is it. And every time I look at images that ChatGPT creates,
Did it work?
It works. No, it doesn't work. It does just work for this. Anyway, I think it's funny because it's obsessed with South Dakota. And I don't know, I did like a bunch of iterations of this, and every time I would do it, it was like, more South Dakota. South Dakota. So this doesn't really sum it up quite. Unfortunately, ChatGPT cannot really solve this problem for us, so we have to do it ourselves. Okay, so the basic question that we set out to answer this kind of body of work is, how does climate change affect the abundance of fish in Midwestern lakes? And I don't need to tell this crowd, this part of the country has quite a lot of lakes crammed into a relatively small area. So we're interested in not just like broad regional predictions of like, in general what's going to happen to fish in Wisconsin, but more lake-specific predictions looking at the variability among lakes. And so on its face, unlike me writing this grant back as first starting out as a professor, was like, well, this is a like relatively straightforward problem, right? This is a species distribution problem. How are species going to change with climate change? We know a fair bit about how climate change affects aquatic systems. We were particularly interested in temperature and warming. We know that if water gets warmer, that can affect the habitat suitability for species with different thermal tolerances. And there's quite a large body of work on projecting, using species distribution models, how the species are going to change in response to environmental change and temperature change across the landscape. So like, okay, let's do this for fish in lakes. It'll be easy. I never actually thought it'd be easy, but so then when we dig into it, though, it turns out there are some nuances that are, I think, important to bring in when doing this kind of work to make the predictions more realistic, more useful for people who might care about them when we're thinking about freshwater fish communities in these lakes. So when thinking about how these communities are going to respond to climate change, we're usually interested in more than just presence-absence. So fisheries are managed for things like growth and abundance and size structure. People who interact with these fish communities, either as recreational anglers or as tribal fishers, care not just about is the species there, but how many are there and how big are they and can we catch them. So we're interested in abundance. We're interested in the abundance of multiple different species that live together, oftentimes interacting in these communities. And we want to know how these species are going to change in lakes that themselves have variable temperatures from surface to bottom and can respond really differently to climate conditions, even lakes that are right next to each other. And so we want these high spatial resolution kind of lake-specific predictions for the abundance of multiple species across the landscape. These are not necessarily like unique challenges to this system, but just the ones that we were facing in a highlight today. So when trying to ask this major question of how will fish communities in this region respond to climate change, really kind of broke out into a number of different questions, which have really turned into like a number of different projects along the way. looking at how lake temperatures are going to respond, how to even start to index the abundance of these species across lots of lakes from lots of states with different monitoring programs. How do we predict changes to temperatures that are maybe outside the realm of what we've observed in the data that we have, and how to do this for multiple species concurrently. So I'm going to walk through these kind of 4 different things to get to the end of like, what's the actual answer to the question that I posed, but I think all these steps along the way have provided some kind of unique insights, and I might use it a little bit of like sort of advertising, branding of like, hey, we've also produced a number of like products along the way that potentially could be useful for other researchers or people who care about this kind of stuff. Okay, so we'll start with thinking about how lake temperatures respond to past and future climate. This is bringing me back to my like old Wisconsin days. A lot of this research, like this body of work for me, started when I was doing a postdoc here at UW-Madison in 2013. And when I went to Jordan Reed and Luke Winslow, who were at USGS at the time, I was like, you know, we're thinking about how fish are responding to climate. Can anybody tell me like how do lakes respond to different temperatures? Can we do that across the landscape? And they were kind of like, well, we can, but like not really at that scale. And that set off a decade of research since then. So we started by asking, you know, can we just use air temperature as a surrogate for lake temperature? They're obviously very correlated. Hotter areas have hotter lake temperatures. Maybe that's a place to start. And you can do that, and that can get you a lot of answers. But there are some limitations. So this slide is from Jordan Reed back when we first started doing this work to show that the correlation between temperature in the air and temperature of lakes, even surface temperatures, does start to break down when you get to higher temperatures because of increased evapotranspiration in the surface water of lakes. So at a certain point, increased air temperatures are not reflected in the increased temperature of the surface of lakes because they just start evaporating more. And then, of course, there's a lot of variability around that relationship, too. So that was one issue. Another one is lakes themselves can have variable temperatures, either across the landscape because of different things like different water clarity, so they absorb temperature differently. Different tree cover can affect how they respond to air temperature. And then within a lake, if they are deep enough, we can have vertical temperature profiles like this, with warm water in the top, cold water on the bottom, transitions in between, that just looking at air temperature might not capture. So we set out to say, well, maybe we just need to get observed temperature from lakes, but anybody who's worked on lake temperature knows if we want to look at long time series of like how are things changing over decades or more, long time series of observed lake temperature data are pretty rare. We don't have weather stations kind of scattered across the landscape in the same way that climatologists who work on air temperature do, so there are places that monitor lake temperature with some regularity, but not usually at the duration or resolution that we might be interested in. But when you start pulling together kind of all the temperature data that we can get across the region, we find there actually are a lot of temperature observations across this landscape. We just might not have like long time series in individual lakes. And so we wanted to kind of leverage those data that did exist to try to build models to understand how lake temperature responds to climate and air temperature conditions. And so over the years, we've used multiple approaches for modeling lake temperature. I'm not going to go into all of this today because I want to talk about fish too, but we've done everything from process-based models that are really based on like first principles of thermodynamics, machine learning models that really are agnostic to the process and just use the data to build predictions, and then some hybrid models that kind of take a little bit of both, with varying degrees of success across different scales. So here's one of those product advertising times. We have produced data sets that are freely available using each of these approaches, again, at different scales. So we've used this hybrid modeling approach just on Minnesota lakes, where we had the data to do it and where we had some specific questions about both thermal and optical habitat. Machine learning models for predicting lake temperatures, just surface temperatures for the entire CONUS. And then process-based predictions, which give us whole water column profiles for lakes in this region. And this is what I'm going to use and talk about today. And for these, we've done both hindcasting into the past and forecasting into future temperatures, both of which will be in use today. So we, together with USGS collaborators, have taken these thermodynamic models of lake temperature that bring in information about the lake itself, about the atmospheric conditions, about the surrounding tree cover, all these things, and applied them to about 11,000 lakes across the region. And again, these produce daily depth-specific temperature profiles. We've done that for hindcasting, kind of what I call contemporary conditions, and then projecting into future conditions for all of these things. And I'll tell you a little more detail, but because we're here with like climate science people, just want to point out, sometimes I skip over this and I'll really gloss over most of it, but just want to acknowledge that we use future temperatures are driven by just one emission scenario, and it's the high-end emission scenario, RCP 8.5, and 6 different global circulation models. That we, for what I'm going to show you today, we're taking the median across all of those GCMs, but we've tried a number of different ways to manage the variability that come out of those GCMs. So when we do this on the lakes in our region, we get these daily lake-specific profiles. We can summarize those in any number of ways, some of which might be more or less relevant to fish communities. The one I'm going to use driving the models I'm talking about today is July surface water temps. And this map just shows lake-specific predicted increases in July surface water temps by the end of the 21st century. But just want to take a moment to point out there's all kinds of metrics available. So in addition to the daily profiles, we produce something like 100 different annual metrics related to water temperature at the surface or the stratification or the bottom water, different seasons, et cetera. And so we can look at things like loss of winter days across the region. It's just one of the hundreds that we calculated, and see some interesting patterns there that we might be interested in pursuing and linking to fish community change in the future. And we're working, a postdoc in my lab right now is working on taking this sort of multiple metrics of lake temperature and combining them in some multivariate space to understand more than just summer surface temperatures, which are important for defining the thermal regime of fish, but trying to bring together multiple metrics. And one thing he's done is come up with these lake classifications based on their thermal regime that highlight kind of the latitudinal differences, but also some of the variability in some of these transition zones where we have lakes right next to each other that have pretty different thermal regimes as it relates to, say, length of growing season, heat in the summer, length of winter, things like that. So, but for now we're just going forward, the results I'm going to talk about are based on July surface temperatures. Okay, so let's just say we did that, got the lake part done. Next big part of this approach is understanding how do we measure the abundance of all these fish species that we might hear about that have been monitored using diverse monitoring gears and times of year and ways of doing surveys across a whole bunch of states and other agencies. So this has turned out to be like the biggest portion of the work for doing this project. Anybody who's done a big data aggregation type endeavor is probably sympathetic. And so there's people in my lab still working on this now. We've been doing it for probably 4 years, compiling fish monitoring records from state and tribal natural resource monitoring agencies who go out and monitor the fish communities in these lakes as a part of their kind of day-to-day business. So we've been in contact with all these agencies kind of repeatedly, iteratively, to understand, first of all, let's have the data, but more importantly, like how are these collected, which surveys are useful for indexing the abundances of which species, all that stuff. So we've ended up with currently close to 60 million fish from over 400,000 surveys and 8,500 lakes in the region. This is not publicly available yet, although we're sharing it with basically anybody who asks to beta test it. And our hope is by the end of this academic year, we'll have our kind of workflow and data released so people can use this for any variety of purposes. And this is just kind of for the fish people in the audience. We do recognize, so there's all of these different gears that people use to index the abundance of all these species. Within and across states, it's variable. And figuring out how to take information from one, electrofishing survey and link it to information from, say, a gillnet survey that is challenging and not necessarily straightforward. So we have statisticians on the team who have been working with us this whole time to develop models that account for differences in catchability of these different gears when we're estimating the relationship between the amount of effort that was put forth and the abundance of the species. And so we do use a subset of gear types and estimate that catchability term for the different gears within a state to try to make them more comparable. And this is another one of the product advertising. So in this talk, we're just focusing on a subset of 8 species that are of interest for this, of special interest for this project. And they were the 8 species that we went out and asked agencies for. There are 8 species that are, kind of harvested fairly regularly, plus Cisco because they're cold water fish. But in doing so, some states gave us like everything that they had. So we do have kind of whole community surveys with lots and lots of species from some of these states. And I also wanted to show off that I made the tops of the graphs the color of the most appropriate state school because that was like a really big moment for me. And yeah, Michigan is green. Michigan is green and white in this. Okay, so we've got relative abundance of a bunch of species. We're focusing on 8 species. And we've got lots of surveys that span a decent time frame for most of these. You probably can't see it at the bottom. These lines are just like the time series from individual lakes for this species. So, and this goes all the way back to, it starts at 1940s. But most of our data are actually from the, like, I would say, mid-80s forward. The data density is a lot higher in that region. And these species are put in order of their thermal tolerance. So we have these 8 species that are of significant interest to management agencies, and that also span a really big range of their thermal preferences. So from the warmest water is preference is the bluegill down to cold water Cisco, and a number of species in between that are cool water species. And so we have species that occur together in a lot of places. We're interested in what's happening with their abundance, and they have really different thermal tolerances. Okay, so that was the part about how do we get the abundance of different species collected by different monitoring programs. And this thermal preference idea leads into one of the big questions we've been working on over the past couple of years. How do we predict the responses of species to temperatures that might be outside the range of what we observe in the data? So if we're projecting into future climates, we might be projecting water temperature regimes that we don't really have an analog for, and so it's hard to say what a species is going to do. So in the kind of traditional statistical species distribution modeling approach, which I have done a lot of myself, the general way that this happens is take environmental variables across a landscape, including temperature, develop a relationship or quantify the relationship between those predictors and the abundance or the presence of the thing you care about, usually using some kind of statistical correlative model. And then you can predict abundance into unsampled spaces or unsampled times using predictions of those environmental variables. So I led a paper that was work that I did here in Wisconsin, looking at basically using this approach for walleye and largemouth bass in Wisconsin, and doing this kind of correlative approach where we can say, okay, we observe this relationship between temperature and the probability of having a high abundance of these species. And in this case, we saw, largemouth bass were more likely to be abundant at high temps, walleye were less likely to be abundant at high temps. But this was just based on correlations observed in the data for the most part. And the question becomes, if we're going to project these really far forward into the future, what do we assume is going to happen with those relationships? Are they just going to stay on the same path? Probably not forever. But how do we determine what we think is going to happen when we move outside that range that we observed before? So we've been exploring this idea of incorporating information of individual thermal tolerance into our statistical models. And so the idea here is that poikilotherms, like fish, so cold-blooded species basically, generally have a thermal performance curve that looks like this, where there is some temperature, the thermal optimum, where some metric of their performance, whether it's growth or metabolism or something like that, is optimized. There's a temperature where they're like really rocking and doing the best. There's also a critical thermal maximum above which we don't expect them to survive. And the curve looks like this, and it's not as clear in this picture as maybe I want it to be, but this curve is asymmetrical, so the steepness of the drop to the bad place is steeper than the rise up to the good places is a bit more. And that's just an important part of this curve. And these are generally measured in the lab on individuals, and the metric of performance can vary. A lot of times it's growth, but there's all kinds of like metabolic measures that people do in the lab on individual fish to quantify this thermal performance curve. And then there is evidence that those individual-based performance curves, based on physiological function of the organism, relate to population level parameters like, let's say, abundance, and that you can use these individual curves to tell you something about how the population is going to do. Okay, so that's taking a mechanistic response to temperature and trying to use that to inform us about how a population is going to respond to climate change. However, building a fully mechanistic model at the scale that we're talking about is pretty challenging. It takes a lot of data measured at a really fine scale, and then scaling it up to like a different kind of temporal and spatial scale and resolution. A lot of uncertainty, a lot of data needs to go into that, and it might miss some key environmental interactions that we might be interested in. And so something that we've been pursuing is developing kind of hybrid models that do a little bit of both. So we've been working with these physiologically guided abundance models that is a data fusion approach that takes both field-derived information on species-environment relationships and also a mechanistic understanding of that physiological performance curve, puts them together when predicting future abundance responses to temperatures, especially those that are getting out of the higher end where we might not have a lot of data about what is going on. So we use literature reviews to put the P in the PGA model. So there's a lot of papers out there examining these performance curves for different species. So we gather that information. One thing that you might be able to see if you have good eyes on this figure, I can't tell if you can see it or not, is so these are the thermal performance curves for each of our 8 species, colored by their thermal tolerance. But for each one, there's a whole bunch of lines behind it, like fainter lines. So if you do this literature search, you'll find, one person does a study in the lab, they find one thing, and another person does a study in the lab, and they find a slightly different thing. So we brought in a bunch of those values and incorporated that uncertainty in the thermal performance curve into our modeling approach. So we drew from different distributions of this curve when making our projections. Anything else I want to say about this? On here you'll also see, just as like small box plots, the range of water temperatures in our region for like present day, mid-century, late century. And you can kind of see how that tracks or not with the thermal optimum of these different species, from the cold water, which is already pretty far down here, up to the warmest water, bluegill, which still looks like it's pretty much in a good range, even in late century. Okay, so there we go, done that one. The last part of the challenge that I saw as approaching was how do we predict the responses of multiple species that live together, potentially in the same lake, concurrently. And this is an issue that I've been grappling with, since my time in Wisconsin, like first starting to look at these, like I showed you that bass walleye figure. I know that there are people continuing to work on this issue to this day who grapple with, if we have, the abundance of one species going in one direction, like largemouth bass are increasing, and at the same time, in the same lakes, we see another species like walleye decreasing. How do we incorporate both of those things into our understanding of what's going on? And historically, at least, there's been a tendency, and this is just like based on human cognition, to attribute the concurrent trends as maybe a causative relationship, right? And that comes out even in some of the regulations or things that people talk about, how to deal with this perceived problem in Wisconsin of like, maybe if we get rid of bass or harvest more bass, then like the walleye would come back. Because if one's going up and one's going down, maybe we take the one that's doing well out of the system, that'd be good for walleye. And there's been a lot of research done on whether this can work or not. And even a lot of, in statistical models, kind of a temptation to say, well, let's use the one species as a predictor of another one and see if that's like a good, strong relationship with the data. But I would argue that doing that kind of mixes up some ideas about correlation and causation that we all know to be true, but sometimes have to be reminded of. Like lots of things can be correlated in time for a lot of reasons, right? This is my favorite website. I love plugging things off of this. I've been doing this for years. This is a new one. Popularity of the first name Johnny, burglaries in New Hampshire. So things can be correlated just by random chance, right? And that's the idea behind this website. They have a new feature now. They'll ask ChatGPT to give you an explanation for the correlations, which can be funny. But also to me, it's interesting because I love to show these figures when I talk about correlation and causation and how to deal with it in statistical models. And really frequently, if you show people a graph like this from the website, spurious correlations, that's only job is to pull data that are randomly correlated and have no causative mechanism, people can come up with an explanation of why they could be causally related. That's just how our brains work. And correlation doesn't not mean causation. Like things can be correlated because they are causally related, but they can be correlated for other reasons too. So one of them is random chance. Another one is they're responding to similar conditions. They're both living in the same environment. They might both be experiencing the effects of that environment and responding, either similarly or opposite ways because of their environmental tolerances. Anyway, that was a long rant, and I'll stop now. So one way to deal with that is to use joint species distribution models. So to model the abundance of multiple species concurrently. And this allows you to ask questions about, okay, if we estimate the relationship between environmental predictors and the abundance of all these species, after you've done that, how much residual correlation remains between these species? Are they still more, positively or negatively more correlated than you would expect, given what we think their environmental tolerances are? If they are, either positive or negative, that doesn't necessarily mean there's a biotic interaction. It could just mean there's some other environmental predictor that they're both responding to that you haven't included in your model. But it can be a sort of hypothesis generating tool of like, look, we included all these things we know affects the abundance of these species. Once we've taken that out, are they still correlated? And if so, like, what could be going on there? Is that biotic or is it something else? And another thing that these models do, which has been shown in a bunch of papers by Jim Clark and his lab, this is for tree species, but the predictions of the abundance of a community is more constrained when you use these joint models than when you use independent predictions of each species individually and stack them on top of each other. So this paper that he did and a bunch of work from his lab showed for tree species and forests, if you just predict individual species responses to climate change and then stack them on top of each other, you get really unrealistic predictions of like the amount of biomass that a forest patch could support. But by doing them jointly, you impose some constraints on, these species don't live in a vacuum, there are multiple species together, we need to account for that when making predictions of the whole community. Okay, so we're going to do that for fish. We can build these joint species distribution models, again, to estimate the relationship between environmental predictors and all of the species of interest, and then look at how much, if at all, do the abundances of these species correlate to each other once we've taken the effects of these environmental predictors out. So this is from a 2020 paper where we did it for like whole fish communities in Minnesota, and then I'll show you the results when we do it for the species of interest that we're talking about with better models now. Okay, so these are the predictors we use in our model. The environmental predictors in addition to temperature that we want to account for relationships or estimate relationships between species and all these predictors. These were chosen in large part based on data availability. What can we get for all these lakes across the region, but also some kind of known or hypothesized drivers of fish abundance. Most of these come from the Lagos database, but some of them were compiled from going out to individual sources. Like, for example, water clarity. We got those from individual stated sources. Okay, so those are our predictors, and we put this all together into a phrase that rolls off everybody's tongue really easily. These joint species physiologically guided abundance models. And we use those to predict the abundance of multiple species across the landscape to changing temperatures, the thing we wanted to do from the get-go. And there's a new component that a grad student or former grad student of Ty Wagner's at Penn State has incorporated to, is we also estimate spatial correlations. So it's actually a joint species physiologically guided spatial abundance model. Anyway, there's a lot of parts going on in this model. but we estimate kind of non-independence of lakes that are close together in space as well. Okay, so those are all the parts. We've produced a bunch of tools, and I think found some interesting things along the way in producing these tools. I guess we're done, right? Like, I was hoping I'd still have time to like tell you about the results, and it looks like I do. Okay. So, we've got multi-species, we've got physiologically guided, lake-specific responses to future temperatures. And here's what we really want to know. How do we predict that these species are going to change in abundance? And then we're also interested as sort of a secondary thing at looking at the probability of abundance going to zero, basically, of extirpation of some of these species. Okay, so it's hard to know exactly how to visualize the results of so many species across so many lakes. So I will try my best to walk you through some of these figures, and then you can ask me questions, and hopefully I can answer them. So a lot of these results are going to focus on percent change. Because we had to account for abundance measured by different years in different places, we can't say like, oh, there's 100 fish in this lake, and now there's going to be 150 next year. So we're looking more at percent change, so we can make those predictions comparable across years with different catchabilities. And we have species ordered again in order of their thermal tolerance. So bluegill are the warmest, Cisco are the coldest. Okay, and these distributions are distributions across all the lakes in the data set. So like 11,000 lakes. How much do we predict the abundance to change in an individual lake by mid-century and by late century? Okay, so a lot of data in here, a lot going on. What can we highlight? Well, these three species with the warmest thermal tolerances, these three species are all predicted to increase in relative abundance in more than half the lakes, even by the end of the century. I should have put a black line at 0, because then it would be easier to see, because you probably can't see it. But there's 0, and these three species, most of the lakes, they're predicted to increase in abundance by mid-century, and still more than 50% by the end of the century. So that kind of makes sense. These are the warmest water species. They're going to, in a lot of places, respond positively to warming. Not everywhere, though. We'll come back to that. All species have some degree of negative response, some number of lakes where they're predicted to decline in abundance. So here I sort of manufactured a 0 line for you. And you can see, again, it really tracks in order of their thermal preference. And by late century, Cisco have a lot of places where they are predicted to be past that CT max and have 100% decrease in abundance. Okay, so here's another way to look at it. This is a map of the percent change. This is end of century. This is late century, so like most warming. And I guess we've reversed the order. So Cisco, coldest water, bluegill, warmest water. It looks like a lot of red, so it's like, oh, everybody's dying. But when you actually go and look at the species with cool water preferences, and especially warm water preferences, you'll see a mix of places where they're predicted to increase in abundance and places where they're predicted to decrease in abundance. Given the color scale and the latitudinal range here, it really looks like, oh, it's a lot, blue in the northern part and red in the south. If you zoom in, there is quite a bit of lake-to-lake heterogeneity in these kind of middle transition zones, but it's harder to see in these whole region maps. So let's talk about Cisco, the coldest water species in our data set. We do predict extirpations in about 25% of the lakes. They have a high probability, like over 90% probability of extirpation by the end of the century, with a big asterisk on it that I'm going to come back to when I say what our next steps are. Again, for the first iteration of this model, we are just using surface temperatures. So we know that Cisco will use deep waters of the lake where it's a lot colder. We include depth in this model, so that does account a little bit for deeper lakes have a not as strong of a response to that temperature. But we are not accounting for the depth heterogeneity of lakes in this iteration that we are working on. And so we know that this is kind of a dire, worst case scenario. For lakes that don't stratify, or if the entire bottom one of the lakes did not have any oxygen in it and we couldn't access it, this would be the prediction, but we know that's not true, and we're working on it. And then I want to touch on our friend the bluegill, the warmest water species, and point out the influence of these thermal performance curves. So if we just based our predictions on kind of the relationship between the abundance of bluegill and temperature in the modern day, present day data that we have, they respond positively to temperature. Warmer is better. And so as things get warmer, we would predict higher abundances. But you can see the impact of incorporating that kind of downturn in the physiological curve. There is a point at which the temperatures get too high. And we don't necessarily see that in the data that we have now, but we're predicting it based on thermal performance curves, especially in the far southern part of our region by late century, where there are places that are increasing in temperature above the critical thermal max even with blue go. That's all I wanted to say about that. Yes. Okay. So species dependencies, I know this is hard to read because I just pulled it out of the paper, so you just have to trust me when I tell you what those numbers are. So these are those residual correlations among species once we've accounted for their environmental preferences. So there are only 5 species pairs where the credible intervals of this correlation, this residual correlation, doesn't overlap 0. And they're all positive. So they're all more about, like, positively related in their abundance, more so than we would expect given their environmental preferences. To me, that suggests there's just other things about the environment we aren't measuring that they both like. Not that they're necessarily supporting each other, but it could be either one. So this I won't read into. These are the species that do have a positive pairwise correlation, residual correlation. And if we look at our friends, the walleye and largemouth bass, where are ya? It's not significant, strongly, it overlaps 0, but it is slightly positive as well. So we don't see any negative correlation in the abundance of these species once we've accounted for their different environmental preferences. Okay. And here are those environmental coefficients. Probably should have put this first. There's nothing like super novel or like eureka moment about these. We're showing relationships between environments and the abundance of these species that we kind of mostly predicted, like, you know, well, I like bigger lakes, like we kind of already knew that, and that comes out in the data. Cisco don't do as well in highly developed landscapes, but they do like clear lakes and deep lakes. So that's the kind of thing that I don't see this as like a result that needs a lot of interpretation, but just it is being accounted for when we make predictions. Okay, so this is what we have done over the past 4 years to try to understand this. There's still a lot of work to be done. Obviously, I've pointed out a number of uncertainties and things that we might want to improve upon, like incorporating the depth-specific temperatures and oxygen availability when predicting the abundance, especially of fish that prefer colder water that's going to be available beyond the surface. But I think this is a good first step to understanding how climate change is predicted to change the abundance of fish across Midwestern lakes. And just want to give a shout out of capturing the range of like variability in these responses and trying to understand interactions between physical processes and biotic interactions and agency monitoring data has really required a lot of collaboration and interdisciplinarity and kind of like creative problem solving of what are we going to do? We know this is a problem and what can we do about it? And it's obviously a work in progress, and I was joking with the mission accomplished because basically every decision that we've made along the way has been like, well, this is better than not doing anything, but still has a lot of work to do. So maybe that's job security. And yeah, we're continuing to work on this in a number of different ways, and I would love to talk to you all more about how to make it better and useful for your purposes. And I'd like to end as we began. This is what I got when I asked ChatGPT, the paid version, by the way, for a map of the Midwestern United States, colored by temperature. And again, like its obsession with some of these states is just really confused. Like it loves South Dakota. It does not appear to like Wisconsin very much. It doesn't know what's here. Maybe that's something. Maybe you got a little colony building in Tennessee. I don't know. But anyway, don't rely too much on ChatGPT to make you images or look at them before you move further. Okay, so that's all I have. So I'll take any questions.
Great. Well, thank you. Are there some questions for Gretchen? I'll let you call them, people.
Okay, I think I saw Alex's hand first.
Since we just had a meeting on this like an hour ago,
Oh, good.
have you tried including recruitment in the physiologically guided models as like recruitment for walleye plus abundance of all the other species?
No, but I would like to.
I figured, I was surprised it wasn't on the next steps.
Yeah, I probably could have put it on the next steps. Yeah, we'll have to deal again with like different geotypes and ways that people monitor recruitment. This is probably a conversation for another room, but people in Minnesota do not agree with Wisconsin's method of quantifying recruitment at all. And we don't have the data to get the measure of recruitment that Minnesota likes from the Wisconsin data. So like, yes, we could, I think that is probably the next step.
But statistically, within that guided framework, there's no reason not to do it, right? You're still just looking for environmental associations that predict abundance for one species and recruitment for another species and interaction.
Yep, yeah, I think the thing statistically we've gotten hung up on a little bit is that recruitment of one species, say walleye, if you also have adult abundance of walleye, they're not, they're correlated in a somewhat different way because one is the babies of another one and grows up to be the other one. So figuring out how to account for that like multi-stage, not just like we have two species that are living together and measured together, but like these are actually the offspring of / future adults of a species. There are methods to do that in this framework, but we have not yet implemented it. Yeah.
Well, kind of jumping off of that, wondering, I know we've talked about this a little bit too, like how much you think or have considered how much stocking might screw up some of your environmental relationships. So essentially we have the DNR dumping fish where they really don't want to be, but then you have that abundance in a place that then might add some bias or more uncertainty into those environmental relationships when you go through the species distribution model.
Yeah, for sure, that's true. We have tried to get stocking data together to incorporate that. As you know, I think we just talked about this in a meeting last month. The data are not always very available. Basically, my conclusion is we need another data aggregation project to get the stocking data from all these places in a format that would even be potential to be useful. So yeah, that's certainly something that is obscuring environmental relationships, and I guess I haven't thought about if it's like by seeing the result. I sort of have in the back of my head that like you can't really stock your way out of a really terrible situation. Like if it's really unsuitable for a species, no amount of stocking is gonna increase your abundance because they'll just die. Which gives, that's just a story I tell myself that makes me think that broad scale conclusions are probably still true, even though it's gonna obscure some of our relationships, but I think wrong. Yeah, Ola.
What's captured in the thermal performance curves, is that for adults or juveniles? Is it growth or is it aerobic scope or?
Yeah, good question. It's for adults. Oh God, I have to go back and make sure that's 100% true. I think it is all for adults, and it is mostly growth. But we did include curves of other physiological measurements as well in that sort of like range of curves that we draw from, but most of them are growth.
That's just a literature bias, right?
Yeah.
Most of
Yeah, that's
most of our measurements are bioenergetic studies.
right. Yeah.
More questions?
Just bad news for Cisco overall. I'm wondering how far north in latitude we're gonna see, like, we'll still have Cisco. And do you know if there's any work out of Canada that's similar?
Good question about Canada. I mean, we still see, even with Cisco specific, like better models that incorporate some of the depth stuff, like broad swaths, especially like along the Granite Shield part of like this part of Minnesota, where it's pretty robust even to extreme predictions of warming, and parts of the UP and northern Wisconsin too. But we also see some of these like bright spots that don't seem like they should still be good for Cisco, but where they're still rocking along, that I think provide some interesting fodder for trying to understand more of that variability. Like, it doesn't seem like they should be suitable, but they're still living here, so what's going on? Not to say that we're not losing them in a lot of this zone, because we are in Minnesota too, but Still rocking along. Round your waters.
Maybe I'll ask a question too. So, so this is really, thanks for the talk. This is really predominantly focused on temperature here,
Yeah.
right?
Yeah.
And then I'm sure you've thought about and maybe incorporating a lot of the indirect feedbacks through temperature, which can have implications here. But how are you, so is this like summer season average temperature? Is this annual temperature increases? Because, again, I don't know anything about fish, but I would guess that, 1 month of really hot temperature versus, versus a summer that's warm, and those summer averages are the same, will probably give you a couple, different outcomes, right? So
Yeah. Yeah.
how does that work into this?
Great question. So this model, and I feel like I'm now feeling like shame about all the things that are not done in a way that would be more robust.
It's easy to poke holes,
Yeah,
right? So,
and like, truly, like every step of the way, we're like, how are we gonna incorporate physiology? Like, okay, now what temperature should we use? So, the temperatures that are used in this current iteration are just mean July surface temps.
Yeah, okay, you did mention that. Yeah.
And for the projections, and for the building, they're like averaged over a 20, 30-year period. So it's like the average increase in July surface temps from present day to end of century.
Yeah.
So
Yeah.
it is not capturing, a heat wave that's gonna come in and cause a fish kill or something, because it's like, what is the temperature over, the July temperatures over a 30-year average? Are
Yeah.
they above the
Yeah.
critical thermal max or approaching it?
Yeah.
I think I am really interested in getting better understandings of more nuanced understanding between temperature and rainfall and fish abundance and how they respond, but we have not yet.
Okay, great, thank you.
I wish we could just say, here's the
Yeah.
daily profiles all the way, like, and not, find the relationship. I guess machine learning could probably do that.
Maybe so. Any more questions? Anything online? Great, well, let's thank, yep, no, we'll
talk about it.
snug it in. Are you sure?
Yes.
Okay, all right, well,